Good morning, everyone who's just joining us. Welcome to a MassMEDIC webinar. We're going to give everybody just a second to jump into the Zoom webinar before we get started. So just stick with us and we'll get started in just 1 minute. Always thought we should have welcome music and we still haven't done that yet. Maybe at some point. I would probably go 80s versus anything else, but just give people another 25 seconds. Okay, fantastic. It looks like we've got a good group. A couple folks are still trickling in, but we'll go ahead and get started because we really encourage a lot of time for questions at the end. So let me get started by saying welcome to this MassMEDIC webinar, and I really appreciate you joining us on this lovely morning here in Boston. It's a little overcast and cloudy. I'm Rachel Robinson. I'm the Chief Operating Officer at MassMEDIC, and it is honestly my honor to be able to join you for these webinars. I learned so much in these discussions, and I know that this one is just going to be an incredible opportunity for you to learn from our excellent guests from Lumicell and Optikos, and I could not do this without them. So please join me in thanking our wonderful sponsors and our speakers. You're going to learn a ton from them today. Just a few quick housekeeping notes. You are encouraged to ask questions. There's a Q&A box at the bottom. I know we've all done this for many years. You've all been on Zooms before, but there's a Q&A box at the bottom. We highly encourage you to put your questions into that box, and we'll answer those at the end of the webinar. Also, this webinar is being recorded, so you will receive a copy. So don't feel if you've missed something, you've got to take copious notes. We will send you the webinar afterwards, and our presenters have also agreed to share the slides. So you will be able to review all of this information at the close of the webinar. So with that, I'm going to kick things over to Dr. David Imrie and say, please take things over and start us off on this wonderful program.
Very good. Thank you, Rachel. I'm delighted to be here. I particularly want to thank our client Lumicell for agreeing to join us on this webinar. It's very helpful to have a client to help tell this story. So I'm David Imrie. I'm CTO at Optikos. I've been here for coming up on 30 years, and that's not untypical of people's tenures at Optikos. We are a consulting company. We've been in business for nearly 43 years now. And the nature of consulting is such that it's always different. Different things are coming in the door constantly. We get to see things that are happening on the cutting edge of our field. And it's that really keeps our engineers engaged and here for a long time. And that's a great benefit to our customers as well, because you get to deal with a team of engineers who have a great deal of experience. We're located in Wakefield, just outside of Boston. We are a hardcore optical engineering company. We address all the different fields of engineering: mechanical, electrical, software, firmware. But optical engineering is at the core of everything that we do. We offer standard products which largely measure the performance of lenses and camera systems. And then we have a consulting side of the business, which is what we're really talking about today, where we develop products for other people. We can sometimes develop just the optical engine of a product, sometimes the entire product. Sometimes it's a custom instrument for measuring something specific that our standard equipment doesn't cover. And in addition to that, we have a manufacturing arm that undertakes essentially contract manufacturing of devices that are optical in nature. We are ISO 9001 and, more importantly for this audience, 13485 certified. So we're able to build medical devices on site. Customers range from aerospace and defense to medical device and life science, which is what we're talking about today. We're also heavily involved in automotive and consumer electronics as well. So that's a little bit about Optikos. I'll pass it over to Jorge to tell you a little bit more about Lumicell.
Well, thank you, David, for the introduction, and thank you Optikos for inviting us for this seminar, and thank you, MassMEDIC, for also organizing this seminar so we can share our successful story of collaboration between Lumicell and Optikos. To get us kicked off, so a little bit about Lumicell, a little bit about myself. I'm Jorge Ferreira, Chief Scientific Officer at Lumicell. I've been working in this project for about 17 years. It started at MIT, and then we transitioned into Lumicell. And about 12, 13 years ago, we approached Optikos to help us with the optical design, and you're going to hear that story today. So at Lumicell, our mission is to improve how cancer surgery is performed, starting with breast cancer. Very likely, every one of us knows someone that has been directly affected by breast cancer, as about 1 in 8 women will experience breast cancer in their lifetime. Every year in the US, more than 300,000 women will be diagnosed with breast cancer, and sadly, about 43,000 women will die from this disease. For most cases, the first line of defense is treatment with surgery. And there's 2 types of surgeries that women can go through. One is a mastectomy, or a full breast removal. And the other option is what is called a breast conserving surgery, or a lumpectomy procedure, which is just exactly how it sounds. It's a smaller surgery where a surgeon tries to remove just the cancer and try to conserve as much tissue as possible. And about 180,000 women undergo breast cancer conserving surgery every year. However, there is an unmet need for lumpectomy procedures, as there is no tool to determine the extent of tumor within the surgical cavity during the procedure, posing the risk of leaving cancer behind that requires a second surgery with increased comorbidities to the patient, poor cosmetic outcomes, and higher costs. So how did we decide to go after this unmet need? Well, to address these surgical limitations, Lumicell's approach was to develop an imaging agent whose fluorescence is activated by the tumor and its surroundings. What that means is that the imaging agent does not produce any fluorescent signal until it reaches the tumor and its surrounding, when it's cleaved by high enzymatic activity in this region. Its fluorescent signal will be then collected by an imaging device that the surgeons will use inside of the lumpectomy cavity to scan for high fluorescent signal and highlight on a screen areas suspected to contain cancer. Therefore, this will be reducing the risk of leaving cancer behind. The initial concept, which you see a couple of a sketch here on our patent, and then an actual picture of the initial proof of concept, was developed during my tenure at MIT as a postdoc under Munji Wendy's lab, who's a Nobel laureate. This prototype was tested in mouse studies in collaboration with David Kirsch's lab at Duke University. And what you see here is a couple pictures of tumor from these mouse studies. The system was built from off-the-shelf components and then modified at the MIT's machine shop. However, this system served as a proof of concept device, but it required improvement before human use. So we did all the preclinical work in mouse studies. We actually did some work in dog studies. And then when we were ready to move into humans, this device was definitely not a human for human use. So this is where we needed an optical redesign to improve resolution and efficiency of fluorescence excitation and emission collection. We needed a more streamlined, reproducible assembly process. And we needed to move from a lab benchtop device to a clinical ready system to improve cleanability, ergonomics, ease of use. And this is about the time when we were transitioning the project from MIT to Lumicell. We needed experts. We needed the help from experts to improve optical design and refine our requirements. This is about the time when we reached out to David Emery and Steve Fantone at Optikos. And David walked us through the Optikos approach and methodology. And I'm now giving it back to David so he can talk to you about the work that Optikos does. David?
Thank you, Jorge. So this slide shows the idealized development path that guides us in developing medical devices. It's very detailed. I'm not going to go through it all, so don't worry about that. And we don't always adhere strictly to this process. Every client is different. But at the very least, it helps us identify where the client is in the process when they first approach us. And Lumicell engaged us in 2013, and as Jorge described, they had already done a lot of the proof of concept work. They had done the work in mice with the demonstrator units that they had built. But they came to us, as Jorge said, because they needed to move on with the next version of the prototype, something that could survive autoclaving, something that would be ergonomic for a surgeon to use, and something that could go into human trials. And what followed was a series of devices that were progressively refined based on trial feedback. And throughout this period, Optikos served as Lumicell's optical engineering department, so to speak. And only when needed, not continuously, but when there was a need, they would come to us and we would work on the next version of the device. And this arrangement is typical with many of our clients. Optical engineering is a very arcane little niche area of engineering in general. There aren't a lot of optical engineers being produced. There are only a handful of colleges that produce optical engineers. So it's usually not practical for companies, unless they're very big, to have their own optical engineers on staff. So we find that the consulting model in which we serve as a startup's optical engineering department, or even a fairly mature company's optical engineering department, works very well. So having worked with hundreds of clients over the years, we get to engage with customers with a wide range of optics understanding. We don't expect our customers to understand optics. As I said, it's an arcane branch of engineering. And to be perfectly frank, even those who've been practicing it for their entire careers sometimes find it to be surprisingly difficult. But in the interest of serving a small educational function today, I would like to convey just a few points that I've frequently found to be confusing to many of our customers. Whether you bring your project to us or to someone else, or choose to go it alone, I hope that the next few slides will help explain a few of the design trade-offs that you might face. You will hear optical engineers referring to first-order layouts of optical systems. In a nutshell, this refers to the basic geometry of object and image locations, magnifications, and numerical apertures, or f-numbers. First-order considerations do not typically involve seeking out a particular lens design. At this point, we're just trying to establish the geometry of what we're trying to do. And here on the left, we have some basic first-order parameters, and on the far right, we have a list of various design considerations. And all of these applied to the work that we did with Lumicell. And we start with the field of view. In a medical device, we most often image from an object, the patient's tissue, in the case of Lumicell's device, to a camera. So we're imaging from an object to a camera sensor. And the field of view is just the size of the area that we need to image onto the camera. In the earlier versions of the Lumicell device, we were imaging an area that was a little under 1 inch in diameter. And so the field of view is determined basically by the optical magnification and the size of the sensor, the physical extent of the camera that you're going to use. Then comes the question of resolution. And this is one of the most frequently misunderstood parameters. So I'll be focusing on it in the next couple of slides. But basically, we want to know what the smallest detail in the object is that you want to resolve. And there's a difference between detecting and resolving. In Lumicell's case, detection was the most important. If I can give an example of what we mean by resolution, if you think of high tension wires that are strung between pylons in the distance, from a long way off, you can see a single high tension wire. That's fairly easy to see. But that is not the resolution of your eye when you're looking at an object like that. To resolve the wire, you would have to be able to see two wires that were the same diameter separated by that diameter of the wire, and to be able to distinguish those two wires separately. So that's kind of the difference between what we mean by detection and what we mean by resolution. And resolution is determined by the size of the pixels in the camera, the magnification, the numerical aperture, which we're going to be talking about in the next couple of slides, and aberration corrections, which will also come up in the next couple of slides. And then there's the question of depth of field. How far along the optical axis do you need to maintain that resolution? And those who worked with microscopes, and I imagine many people in our audience today have worked extensively with microscopes, you will understand the idea of depth of field. For very high magnification, high numerical aperture microscopes, focus is maintained over only a very small distance. But for a camera system in a patient, you may want to maintain that focus over a much longer depth of field. And those are kind of the geometric optics considerations. There are certainly plenty of others that I haven't spoken about here. And then there are the sort of photonic physical optics sides of the issue as well. Questions of sensitivity, what is the photon budget, determined by how big the pixels are, and in this case, how intense the illumination is, because in a fluorescence system, you're providing the illumination and looking at what you get back. So there's a photon budget, making sure you have enough sensitivity to detect the thing that you're looking for. And then again, in a fluorescing detection system, there are spectral considerations, particularly pertaining to the filters, the excitation and emission filters, looking at how narrow those need to be, and a couple of other things that we'll be discussing in some slides shortly. But I do want to turn to the question of numerical aperture. So in the next two slides really cover the information that I would most like to convey. If you have one takeaway from today's presentation from a technical point of view, this is probably it. Many of the medical and life science instruments that we design or work on involve something akin to microscopy. So I'm going to be looking at resolution from that perspective. And if you've worked with microscopes, you may be familiar with the concept of numerical aperture, but if you have not, then the sketch that we have here illustrates that concept. We're showing a lens element in blue, very simple sketch that is bringing a bundle of light rays shown in yellow to a sharp focus. The numerical aperture, or the NA as we will call it from now on, is simply the sign of the half angle of this cone in air. In another fluid, you would have to include the refractive index, which is the small n that you see there. But in air, it's just the sign of that half angle. And in PowerPoint, we can focus to a perfectly sharp focus. But in reality, there's a physical limit to how sharp of a point you can achieve. And this is a consequence of diffraction at the edges of the limiting aperture in the system. And so we call it the diffraction limit. And the distribution of light at the focus appears as we see in the photo here. Although the outer rings that you can see in this photo have been enhanced for clarity, we are most concerned about the diameter of the bright spot in the center of the photo, which we see is proportional to the wavelength and inversely proportional to the numerical aperture. That is the airy disk. I like to think of this spot as the size of the pencil tip with which we are drawing the image. So a higher NA will produce a sharper image. Here's an example of a 40x 0.95 microscope objective, and we can learn a few things about it from the markings on the cowling. We can determine what the cone angle is. If we apply the formula for numerical aperture, we find that the half angle is 72 degrees, so the entire cone will be 144 degrees. We can determine what the smallest object is, what the airy disk diameter is for a well-corrected microscope like this at, say, 546 nanometers, the mercury wavelength that we frequently use. And it comes out to be about 0.7 microns. And at the same time, we can use the 40x to figure out how big that would be at the camera. So we take 40, we multiply by 0.7, and we get 28 microns. So right there, that tells us that there's very little point in having something like 1 micron pixels on this camera when the smallest object at the camera is going to be something like 28 microns. So it allows us to make a sensible choice about how big the pixels in the camera might be when we use this microscope objective. And a lot of folks, especially those who've taken a few microscopy labs, will have understood a lot of what I've said so far. But when designing an optical system, we saw that picking the NA was kind of a first-order decision. So the question is, why not make all lenses high numerical aperture and high resolution? Well, there are 3 very good reasons. And here's typically the least understood reason. It's cost. Even though you have selected numerical aperture to deliver high resolution, you still have to correct all of the aberrations in the lens to achieve this. The diffraction limit is just an upper bound for how good a lens can be. Actually achieving the diffraction limit gets exponentially more difficult as the numerical aperture increases. I'm certainly not going to dive into aberration theory today, but the simple truth is that shallow rays near the optical axis are more easily controlled than the steep ones at the edge of the cone. So smaller numerical aperture systems typically need fewer optical elements and are easier to assemble. So there are a couple of elements that are really important to understand. The expense comes from, first of all, having to design a more complicated lens. But then you're putting more elements into the lens in order to correct the geometric aberrations. And then you have to build that lens. And that means for a very high numerical aperture lens, you have to align all those lenses very precisely. So building the lens is much more difficult for a high NA system that has to perform to its diffraction limit than it is for a low numerical aperture system. So that's where the expense comes from. You don't get the resolution of the numerical aperture for free. It's just setting an upper bound on how good it can be. The second reason is that high numerical aperture lenses have a much shallower depth of field than low numerical aperture ones. Low numerical aperture lenses, and that's simply a question of geometry. You have these cones, and as you go through focus, you're intersecting the cone. And you're getting a different spot diameter. So for a steep cone, that spot diameter increases very rapidly as you go through focus. You don't need to know anything about optics to understand that. That is just pure geometry. And the third reason is in medical devices, you quite frequently want to put something in the working distance being the distance between the last element and the focus. And in a high numerical aperture system, that distance is much shorter because the cone angle is steeper. If you're trying to increase the distance in the working distance in a high numerical aperture lens, then the elements become very big very quickly. So the working distance is quite frequently a consideration as well. So those are my quick takeaways on resolution and high numerical aperture lenses. The message is basically don't design around more numerical aperture than you actually need, because you're going to be paying for something that is not free. High numerical aperture costs money. It costs more designed elements, more precise elements, and more time to assemble them. I want to turn now to how we go about building optical assemblies and where we get optics from. And this applies a lot to the prototyping, but it also applies to the final product as well. It hardly ever pays to reinvent the wheel, and not every optical product requires a custom optical design. And we don't automatically reach for a custom solution to every problem. Depending on the developmental stage, the performance requirements, the expected manufacturing volumes, there may be off-the-shelf options that can be used. And I do want to emphasize that the use of off-the-shelf components is not confined to prototyping. They can be a legitimate means of procuring the optics for many instruments. So first, there are optical assemblies that can be very useful, and most obviously microscope objectives, right? Those are optical assemblies. Somebody else has designed and built them. You can buy them off the shelf. The photographic lens assemblies would fall into this category as well. The upside of using off-the-shelf assemblies is that the cost is reasonable compared to designing and manufacturing your own. You get rapid delivery. You can get a lot of these things overnight if you need them overnight, if they're on the shelf. And there's a convenient amount of precision that comes with them. The cons and downsides to this is you're stuck with the form factor that the manufacturer of that lens assembly came up with. They have fixed optical parameters. You can't really adjust anything in them. And the performance can be a little mixed. As much as manufacturers of microscope objectives will, they'll certainly tell you what the numerical apertures and the magnification and so on. Very few of them actually sign up to performance specs. So you're never quite sure what you're getting. Certainly some are better than others. There's a bit of potluck. We've certainly measured a lot of different microscope objectives and many of the same type. And there is a distribution of performance in off-the-shelf objectives. Manufacturers aren't saying they're going to meet a particular performance metric. The second place that we get optical components is literally components out of catalogs. There are a few big component vendors. You would have heard of Edmund Optics, Thor Labs, Newport, places like that will literally sell the elements by themselves. Singlets, doublets, mirrors, prisms, filters, that kind of thing. Plenty of components that you can then take and use for prototyping, and as I said earlier, even in the final product itself. And in the case of Lumicell, this is a lot of how we set about developing the early prototypes. The upside of using catalog components is the cost is modest. Again, you can get rapid delivery. You can get these things overnight if they're in stock. And there's a flexibility to the assembly that you don't get with the off-the-shelf assemblies. You can make your own housings. You can determine the form factors for those things. You can determine the spacing of elements and so fit them to meet your specific needs. But on the downside, there is going to be a limited design performance you can get using off-the-shelf optics. You are stuck with the offerings that the vendor has. You can't, specify different sizes or focal lengths, that kind of thing. You're stuck with the fixed focal lengths and sizes that they offer. And the performance of off-the-shelf components is going to be limited compared to an off-the-shelf lens assembly. You're never going to build a high numerical aperture microscope objective from off-the-shelf components. And again, you don't know how well they've been built. There's the actual fabrication performance may not be great, but there is certainly a very convenient source of optics. And then finally, if you can't use those resources, then you have to resort to designing and building your own lens assembly. And that's where a company like Optikos comes into it. Although, as I said earlier, we will absolutely use off-the-shelf components if that makes sense. But then we move into optical design, which is a specialized branch of optical engineering to actually design lens assemblies. But here you have full control over the form factor, the performance of the optics, if they're windows, all sorts of parameters that you might want to put into the optics can be introduced for a custom lens assembly. And potentially for some medical devices, we can introduce molded optics as well for high volume disposable endoscopes, that kind of thing. If you're using molded optics, those all generally have to be custom. The downside, of course, is it's much more expensive than using off-the-shelf components, and the lead times are going to be that much longer. There's a lead time to design it. There's a lead time to have the components manufactured, and then you have to assemble them as well. So a different stage in the process, perhaps. But if you're unable to use off-the-shelf stuff, then custom lens assemblies are where you have to go. I want to talk a little bit about MTF because MTF is at the heart of everything Optikos does. MTF is a measure of lens performance. If you work with us or with other optical engineers, they will talk about MTF. You will see graphs like this. This is the MTF graph of an early Lumicell prototype. Very briefly, MTF is the modulation transfer function. It's a measure of the contrast in the image. And it's runs from 0 to 1, although typically people will talk about MTF as a percent. Drawn on this graph, and you can just see it behind the top green line, there is a black line, and that is the diffraction limit. So for this particular lens, that is as good as the MTF could be. Along the bottom axis, I should have mentioned, we have spatial frequency. So spatial frequency is measured in lines pairs per millimeter, cycles per millimeter. And that goes back to what I was talking about earlier with the high tension wires being adjacent and separated by the thickness of a wire. Those are line pairs, so lines that are certain thickness separated by the same thickness. And as that frequency increases, the resolution of the system is going to drop, not least because the diffraction effects are going to start to blur that image. And we're not always trying to get the diffraction limit. We're not always trying to design a lens to hit the diffraction limit, but we will have certain MTF targets at certain spatial frequencies. And the spatial frequencies we're interested in are usually going to be related to the Nyquist frequency of the camera that we're using. So if you have 5 micron pixels, your cutoff frequency for that camera would be 1/5, which is 200 line pairs per millimeter. Your Nyquist sampling frequency will be half of that at 100 cycles per millimeter. And quite often we're looking at Nyquist over 2, so 50 cycles per millimeter would be a frequency of interest. And then just a couple of interesting aspects of the Lumicell design. Jorge explained that it was a fluorescence detection system. This is an early optical layout. If you work with optical engineers, you will see ray traces like that. This is exactly what this is. This comes from a software package called ZMAX. And it shows the ray bundles emanating from the object on the left and eventually focusing on the camera sensor on the right. There is an illumination path that comes from below that bounces off the beam splitter that you see there, which is not explicitly illustrated in this case. And in an ideal situation, you'd be having sharp points on the left imaged to sharp points on the right. A couple of interesting things in this design is that there was a window in this instrument that gets pressed up against the patient's tissue, so that defines the image plane. In this case, a fairly thick window. And as a cautionary tale, I suppose, always be aware of introducing plano-parallel windows into converging beams. It is always going to introduce what we call spherical aberration, which is illustrated in the graph below there. And the steeper that cone, in other words, the higher the numerical aperture, the more spherical aberration you're going to introduce by putting a plano-parallel window in a converging path like that. I recall when I first joined Optikos in my first couple of years here, I was called to visit a startup company that had built an instrument and had built quite an elaborate prototype of their medical device. It was, I forget exactly what it was, but it was quite an elaborate desktop instrument. And they had a very high numerical aperture, long working distance objective, a very expensive objective that was imaging the sample through a very thick window, maybe 5 millimeters thick. And they couldn't understand why the image wasn't nearly as sharp as they had expected it to be. And they'd gone through the process of this entire design and build without having understood that you can't just put a thick window in a converging beam and not seriously affect the resolution of the image. So there's a cautionary tale. Whenever you put a thick window in a converging beam, be aware that you are introducing aberrations. If it was a custom design, you would totally design out the effects of the window. But if you're putting it into an off-the-shelf component or assembly, it will not be, that design will not be expecting you to put a window there. Other things that we see in here, interference filters, of course, for the emission path and the dichroic beam splitter that we see there. Be aware that narrowband interference filters will change their center wavelength based on the angle that of the beam that intercepts them. So that's something important to be aware of in a fluorescing system. If you have a high numerical aperture going through that, it's going to probably bleed other wavelengths through it. That's just because you're going through more material in that interference filter, and so you're changing the way the interference filter behaves. You will also see things like the paraxial lens on the right there, which is just shown as a line here. In an optical design, when you're just doing first order layout, you may not put in real lenses. You might just put in ideal lenses, which is what that paraxial lens is. So if you're looking at optical layouts and you see lenses that are just shown as lines, or lines that are behaving as lenses, those are probably just paraxial lenses, which are placeholders for real lenses that are coming later. And here's my last slide, just one more cautionary tale. Also, this applies to the Lumicell design. This is another view from Zemax, a 3D view of a slightly different model later on in the design. And this one has to do with folding an optical path. Folding is the term we use to when we're referring to bending the path using a mirror. If you have a converging path, when you put in a folding mirror, it's not going to be centered on the optical axis. You can see the red arrow pointing to where the optical axis is, and it's well displaced from the center of the mirror. Just the nature of that cone angle means that your folding mirror has to be significantly displaced from where the optical axis intersects it. So a couple of cautionary tales there, mostly to do with converging beams, but just some things to be aware of. And now I'm going to hand it back to Jorge to tell you a little bit more about the actual process that they went through.
All right. Thank you, David, again. So while all this development, the optical design was in progress and taking shape, we as a medical device company needed to consider all the requirements and regulations governing this field. We had multiple conversations with the FDA describing the device design, which again, opticals play a big role on providing all the documentation. You know, these ray traces, we use those to present to FDA to model the images, what kind of efficacy result we will have based on this design. And we also had to discuss the clinical development plan with the FDA. Our system, for those that are in the MedTech world, was categorized as a class 3 device, which is considered a high risk. And the reason for this is because the system, the device will be used inside the cavity, an open wound. So the system will be actually touching tissue. But not just only that, the system has software that will tell the surgeon whether to take more tissue or not. So surgeons will be making clinical decisions based on the imaging performance of our system. So we were categorized as a class 3 device. So the regulatory path for our device was a pre-market approval, or what's called a PMA, which has a lot more stringent requirements than a typical 510K, where you have a predicate device. You may not need to run a whole bunch of clinical studies. So for a PMA, there's no predicate device. We had to come up with all the clinical evidence and all the manufacturing, all the documentation that was needed for this device, and not just the device, but also for the drug. Next slide, please. All right, so let me walk you through a high-level timeline of how we went about working with Optikos. So in 2013, we had our initial conversation with Optikos with literally a lens tube design. Then in 2014, they built our alpha unit that supported our IDE submissions to FDA and carried us through the feasibility studies in humans with our clinical collaborators at MGH. This happened between 2015 and 2017. I'm gonna show you, I don't know if you can see these, there's, one of the things that Optikos do, they have rapid prototyping 3D modeling of devices. So I have here a few examples of things that Optikos did for us before we nail down the final design. So later, Optikos took our learnings from the early clinical studies to improve the design for much better usability with clever placement of lenses and mirrors. So all the cables would come out from the back. Originally, we had a cable coming from like 90 degrees out to make it easier for the design, but it was difficult for surgeons to use. So that was feedback that we got from the surgeons. And Optikos incorporated that into the new design. Optikos also designed the main body of the system so it can have interchangeable, what we call imaging tips. And this system was used in a large multi-site clinical study between 2018 and 2019. And the purpose of these imaging tips was to, based on surgeons, feedback from the surgeons, obviously not all breasts are created equal, not all tumors are created equal. So they were asking for us to give them options to accommodate for different sizes of tumors, surgical size, and breast size. So after our larger feasibility study, the design of the system was locked, going into what we call the pivotal or phase 3 study phase, which was then completed in 2022. One thing that I skipped over all this time is that at every single stage, there was a significant amount of planning and testing to verify and validate the design iterations to meet our design controls and IDE submission requirements. And the rigor of the work performed by Optikos really helped Lumicell to generate the documentation that we needed to support our IDE submissions and the benchtop evidence that was necessary to support the expected results from our studies going into the clinical trials. So this leads us where we are today. So we are no longer a research stage company. Our drug and device were approved by FDA in April 2014 through the most rigorous and demanding regulatory path. Our drug was approved under an NDA, and our device was approved under a PMA. And we had to work with both groups at FDA to coordinate the review, make sure that everything was moving forward at the same time, because this is a combination product that the drug needs the device needs the drug. So both of the components had to be approved at the same time. Our current system is in commercial use at about 11 hospitals throughout the US, and we continue our commercial launch. Our drug's brand name is Lumicell, which is an optical imaging agent that produces fluorescent signal at the site of residual cancer. Our device's brand name is the Lumicell Direct Visualization System, or DVS, which consists of a handheld probe that is inserted into the lumpectomy cavity. And this is the part that Optikos really helped us to design, this handheld probe. And a workstation that has a computer that displays the regions suspected to contain cancer to the surgeon. So the surgeon are doing the surgery, they insert the device on the lumpectomy cavity, and they look on the screen. And when there is high fluorescence, the system will tell the surgeon this is area suspected to contain cancer, and the surgeon will go back in and remove additional tissue. We show in our clinical trials that by using the system, surgeons can identify residual cancer that would have been left behind in these patients. We also showed that we can reduce the amount of second surgeries by finding this residual cancer and converting some patients from what is called positive margins, meaning that they might have residual cancer left behind, to negative margins. So this patient would not have to go to second surgeries. I guess to finalize, we're really pleased with our collaboration with Optikos in this project that require a really demanding and complex optical design. David explained some of the details that we had to go through the design of the system. This system produced a successful surgical outcomes, only achieved by a strong partnership between our engineers at Lumicell and the experts at Optikos that led us to this successful system design. And now I'll give it back to David for some closing remarks. David?
Thank you, Jorge, for those kind remarks. I have to say that Lumicell was a delightful customer to work with. They're still a client of ours. We're not allowed to have favorite clients, but Lumicell was a good client. So a couple of takeaways from this. What made this collaboration successful and what continues to make it successful. Something that might be helpful to our audience, whether they partner with us or with somebody else. Partnering early in the development process was important in this case. Lumicell had done the lab work and then brought us the problem of developing the actual product. Getting in early is always helpful. We will jump in at any point in that process, but the earlier the better is a pretty good rule. There was a clear division of responsibilities in this case, as there usually is. We don't get involved in the chemistry. That is not our expertise. That is entirely Lumicell. They have the chemistry and the drugs all locked up. We're only interested in the optical engineering part of it and the product engineering. And that division was very clear. The third point here, and this I can't emphasize enough, the open sharing of information is very helpful. Lumicell shared everything with us. They're an unusually transparent client, and it really makes a difference. Knowing as much as possible enables us to make appropriate design decisions. It can happen that the customer feeds us only the information they think is important, and they may omit a critical detail. And that has happened to us before. There is one notorious case that happened when a customer asked us to design a lens for them, and only when we were presenting them with the assembled lens did they happen to mention that it was going to be used underwater. So that's an egregious omission of a detail, but there are far more subtle things that can happen as well. So don't assume that you're telling your engineering consultant everything they need to know. Rather overshare. And we like to tell people they can never tell us too much. We take confidentiality very seriously, but once that NDA is in place, you should really tell us everything you can about what it is you're trying to do. And last bullet point here, we avoided over-engineering the performance. As I mentioned before, this is not a question of high resolution in the case of the Lumicell device. It's a question of being able to detect fluorescing cells, not to be able to separate two adjacent fluorescing cells. That's not important. We're looking for cancerous tissue that's in the wound, and to do that, the need is a detection rather than a resolving requirement. And I think that wraps us up. I certainly appreciate people's attention for joining us today. I think we have a few minutes for questions and answers.
Yeah, I mean, I would jump in really quickly with one question if I could. Knowing what you know now, Jorge, about how difficult the path was, you went through the most difficult path for a drug and for a device. Is there anything you may have done differently or done differently even in the partnership? Is there anything you wish you knew before you got started that maybe you could kind of share with the audience? And David, you've done this a lot too, so would love your perspective. Are there any kind of tips or things you think it would be helpful for folks to know before walking down that path?
I think David mentioned something that is really important for us, for Lumicell, and it's transparency. Like when we approached them, we told them everything that we knew, and probably even more. Challenges that we were expecting ahead, and just being transparent. Like we're gonna go with a device a design. This is a design that we think we need. And as we went through clinical trials, that device design changed because we got feedback from surgeons. So I think just to reinforce what David said, that being transparent and just this is what we need, and be sure that that design may change as we collect more data. Looking back, I think that something that we could have done slightly different is maybe look at, we started with a very demanding design, for sure. And David, I don't know if you remember this, we came to you and we said, we want to see a single cell. And there were discussions, to David's point, about detection versus resolving. And when we approach and we say, like, we want to see a single cell. And those conversations evolve. And I remember Steve Fantone also being part of those conversations where, going again, open-minded at these, they said, look, imagine the night sky, right? You see a star, right? You don't know what the size of that star is. You don't care. You just want to see it. And that really kind of changed our mindset of, okay, I don't need to know the size of this cell, or there's another cell next to it that it doesn't have the signal. We're trying to look, stars in the sky. So I think going also with an open mind, that what you think is what you want is not necessarily what you actually need. So listen to the experts, listen to the folks that probably have done something like this before, and be open to get, for options and suggestions that you might not have considered before.
Yeah, I think that's very true. So when you're approaching somebody for a consulting job, whether it's in optics, as it is with us, or something else, it always helps to tell them what it is you're trying to do rather than how you want them to do it. Because they probably, we get customers that come to us with all sorts of problems. Sometimes they have already built something and they want to turn it into a manufacturable product, but they really rigidly want to stick with what they've worked on. And that's understandable. And in the case of startups, it's their baby, right? So people bring us their baby and say, here, take this and find a way of manufacturing it. And we'll look at it and we go, is that really how you want to do that? There's better things you can do here.
You can't call that baby ugly, David. You can't.
You can't. So, but we always try to steer the customer towards a sensible decision. In the end, sometimes taking a couple of steps back is going to really pay off in the future. If you're going to push forward with something that's unmanufacturable, that can be really problematic. But we encounter all sorts of different stages of development, and we're happy to work with customers at any point in that stage. But as I said earlier, the earlier the better. If you just come and ask us for some advice early on, we can probably help steer you in the right direction. And
that's fantastic.
Yeah. One of the things we do say, and we have a lot of sayings, we have a lot of sayings, and one of the things we do say is optics is hard. And it really is. It's this weird engineering discipline that is very hard to understand unless you've been living it. And from our point of view, we live optics every day. We look at a problem and we immediately see all the options, all the constraints, and a whole lot of solutions as well. And that's true of any discipline. It just becomes second nature in the end. When you look at a problem, you just have that insight. You look at it differently. Mechanical engineers look at engines differently from how I would look at them, and civil engineers look at buildings differently from how I would. And that, I think, is helpful. I think it's helpful to go to an expert early on in the process and just try and pick their brains.
Awesome. Well, I want to say thank you to both of you for this wonderful presentation. Really appreciate it. It's a ton of information, David. That was very interesting topic and tidbits that you were getting throughout. So I know folks will very much appreciate the slides and the video and the chance to review. So thank you again. And thank you to everyone for joining us. We hope you'll join us for another upcoming MassMEDIC webinar. And you can check out our upcoming events. There are a lot on our website, massmedic.com. And I encourage you strongly to reach out, obviously, to David and the Optikos team. And I'm sure Jorge would be open to taking questions another time as well. So thank you
Rachel,
all. Yeah.
Before we go, a question came through about whether the FDA classify our product as a combination product. The answer is yes. That's a simple answer. The longer answer is we had to submit at some point what is called a request for designation to FDA, where we believe that our combination product, the drug and device, had a primary mode of action of a device. Like the drug is essentially just giving you a signal, and the device is really telling you where that signal is, how much signal there is, and telling the surgeon you need to cut here. So we had to go to the request for designation process, and then the FDA agreed that yes, this is a combination product with a device primary mode of action of a device. So all of our submissions during the clinical development were through an IDE, not an IND, strictly as a device. But the drug group was always reviewing all of our IDE submissions. So this did not mean that we didn't have to meet all the drug requirements. We had to meet all the drug requirements, but everything was being submitted through IDEs. So even our the preclinical safety studies in rabbits, like we were testing the safety of the drug, we submitted those results under an IDE, which is unusual, but that's the way FDA wanted to go about it. But when it came down to the approval application, we were thinking of that, okay, this is going to be a PMA, and then there'll be like an NDA section within the PMA. And the FDA basically at some point said no, and they were not really giving us a good answer, so we decided to submit both an NDA and a PMA. That was actually the cleanest way for us, and we believe that it was also the cleanest way for FDA, because they can just look at the drug as a drug, look at the device as a device, and there were constant communication between the two groups. So yes, it's a combination product, but it's very complex system, because you have an NDA and also a PMA.
They think that explanation was very helpful. I do too. That was awesome. Thank you for clarifying that. Okay, I think we've answered all the questions. So I think this is a great time to say again, thanks everyone, and we'll see you at another future program. Have a great rest of your day.
Right.
Thank you.